So phosphorylation of autophosphorylation of CQSS results in a phosphotransfer to this response regulator LuxU, and then LuxU phosphate will transfer its phosphate group to LuxO. And in the presence of sigma 54, which I'll talk a little bit about later, sigma factors in general, LuxO phosphate will actually activate the expression of small RNAs and this other protein called HFQ. So together, the small RNAs in HFQ will block the central regulator HAPR. Now in Vibrio fischeri, which we've been working with a fair amount, LIDAR is the homolog, and that's, for example, the PMF8 mutant is a LIDAR mutant, which is a homolog of the Vibrio cholerae HAPR. So again, no autoinducer. That triggers this phosphocascade and ultimately results in luxophosphate, which activates a repressor of the central regulator HAPR. And so HAPR's job is to activate protease genes and things like that and block these types of activities, virulence and biofilms and so forth. But the main things that I wanted you to take from this are the multiple layers of transcriptional regulation that are happening here. You've got autoinducers, you've got a multi-component phosphorelay system, you've got small RNAs and a stabilizing protein like HFQ, and you've obviously here you've got another transcriptional activator and repressor as well. This dual role of activator and repressor. So again, it's really sophisticated stuff, and it occurs at multiple levels. So the only thing I wanted you to get from this table, well, overall, I just wanted you to see an array of small regulatory RNAs, their size and who produces them and what their function is. But the main thing I wanted you to see here was just NICF. It's a 109 nucleotide small RNA that has an antisense function for inhibiting OMP-F, messenger RNA translation, as I talked about. So that's the focus of this slide. And so the important concept here is that microbes will regulate large groups of genes, or rather co-regulate large groups of genes. Now, obviously, they can do it at the operon level, where you have a single promoter regulating expression of two or more contiguous genes. But they can also do it at the regulon level, where genes or operons are all co-regulated. Individual genes, and these are genes and operons that are scattered throughout the genome, but they're all plugged into the same circuit, which is great because it allows the microbes the ability to rapidly adapt to changing environments. So one of the components that I alluded to before was a sigma factor. Microbes use these to interact with RNA polymerases. And so they have different kinds of sigma factors that can bind to RNA polymerase and then drag the RNA polymerase to certain kinds of promoters, depending on what's necessary. So one that I talked about and briefly mentioned here was sigma 54 that interacts with LuxO. Another one, though, is another one is sigma 70 that is essential for activating RNA polymerase and expressing genes in exponential growth phase. Whatever the microbe might need to grow quickly. There's another one called sigma S that's for activating general stress response during stationary phase. So the microbe will favor sigma S as the environment starts getting terrible and the microbes stop growing as well. But there's a whole bunch of others, and I had even mentioned this guy previously in the discussion about the regulation and the building of the basal body motor and flagellum, sigma 28. And I had talked about the anti-sigma factor as well that works in conjunction with this. But again, so you can see that there are different sigma factors that microbes use to make their RNA polymerases modular, so they can activate all kinds of different gene expression. The RNA polymerase has the same core structure, but all you've got to do is swap out one part, and then the RNA polymerase becomes more adept at activating certain genes. So one of the big ones, and we've worked with this already in lab, is the cell-cell communication that microbes use. And so it was really common for people to, it's always been really common for people to think about microbes as sort of these planktonic loners, just, like fish swimming alone in a fishbowl. But they're really not. They're mostly very social little creatures, and they're often associated very intimately with one another, whether it's in a biofilm or some kind of slime layer or attached to tissue or something. And so they're not just by themselves. They're very rarely, again, very rarely floating around planktonically in media, unless they're in media in the lab. Usually that's a very artificial circumstance. Most of the time, like I said, microbes are in communities on surfaces, and the way they operate in those communities is to speak to one another through, well, quote unquote, speak to one another through acyl homoserine lactones in the case of gram-negative organisms. The concentration of acyl homoserine lactones allows them to assess their own population density and then turn on or turn off certain genes as necessary. If there's a gene that's not necessary in a high density, then they turn it off. And if there's a gene that is necessary when they hit a high density, they can turn it on. And so again, these are small molecules that can readily diffuse across the plasma membrane. And again, they can achieve fairly high concentrations in the environment, and they can, again, freely move in and out of the cell, and they regulate a variety of functions. And so this table overviews some of those. These are the typical structure of gram-negative type homoserine lactones. So they're N-acyl homoserine lactones. This cyclic ester that you see here is the lactone, and then there's an acyl side chain of various carbon lengths, and then various certain decorations that stick off of it. And again, these are important. The chain length, for example, can dictate the type of language that's being spoken. And some microbes can only understand a 12 carbon length, and some microbes can only understand a 4 carbon length. And the autoinducer we worked with in the lab is a 6 carbon length autoinducer that Vibrio fischeri produces. So again, all kinds of functions, lots of different microbes make them. This is autoinducer 2. This is the more generic one that I mentioned previously in the cholera example, but fischeri has it. Lots of microbes produce an autoinducer 2. What's really cool about this molecule, though, is I don't know if there's another example of boron being important, like in a biological system, but this molecule has a boron cofactor, and so it was a really neat discovery that autoinducer 2 has this really unique structure. It was first discovered in Vibrio harvii, but now we know that E. coli and lots of microbes make autoinducer 2 and have this, as I showed you previously, this system here, this LuxS based system. So LuxS is the synthase that actually makes these triangles, and these triangles are the boron cofactored molecule that I just showed you. But it's not just gram-negatives, that are making these autoinducers. It's also gram-positives, and the primary autoinducer produced by gram-positives, like Staph aureus, are these little cyclic thiolactone type. Basically, they're just little peptides. In this case, it's 1, 2, 3, 4, 5, 6, 7, 8, 9 short little 9 amino acid peptides. And so you can see the cyclic part here. But this is what a lot of gram-positives communicate with. Staph aureus turns on and off lots of virulence genes using this cyclic thiolactone type approach. But there's, plant pathogens like Ralstonia and Xanthomonas that use different kinds of molecules. These palmitic acid methyl ester, methyl dodecanoic acid. So these are additional types of molecules that are made by, in this particular case, some gram-negative pathogens, plant pathogens. But even the yeasts get in on the action. So here's Candida albicans using farnesoic acid. So these unusual acids, and this can regulate virulence and dimorphic transition. Candida albicans is a dimorphic fungus. They can exist as individual cells, like you're used to seeing yeasts, but they can also exist as these filaments. and they're usually in their most pathogenic form when they're in the form of those filaments. So in the case of individuals when they have yeast infections, either urogenital yeast infections or oral thrush, the pathogen Candida albicans is typically using these types of molecules in order to communicate and convert transition in and out of these pathogenic and non-pathogenic forms. So this slide is just an overview of the oligopeptide-mediated quorum sensing in gram-positives. And so the microbes will produce these short little peptides that get processed and then pumped out of the cell by these ABC-type secretion mechanisms, ATP binding cassette secretion mechanisms. And then they have a two-component system usually that recognizes the peptide. Here's the peptide. Here's the sensor kinase, the first component, and it autophosphorylates when it detects this peptide. Then it can transfer the phosphate to a response regulator, and then the response regulator in its phosphorylated state can activate or turn off genes. This is sort of the overview of the acyl homoserine lactone type systems and gram negatives. So again, and here's a little timeline sort of in a non-chorate system, meaning that low cell density, there's just a basal level of transcription and translation happening. But as the cells grow and the optical density of the culture increases, they start to accumulate signal, especially if they're trapped in a test tube or a flask. But this could also occur if they're trapped in a confined space in a host animal as well, kind of like the squid light organ that I talked about in that lecture. Then they transition to a chorate type system where their cell density is high and the amount of acyl homoserine lactone autoinducers in the environment is high. And that can start activating sensors and in turn activating targets. So typically these microbes have some kind of LuxR type homolog and a LuxI type homolog. And so the LuxI is the synthase that makes this molecule. This is the autoinducer signal. We use the pure form of this in our autoinducer luminescence experiments. And so this signal sensor will bind to the autoinducer to make an active transcription factor that can then turn on or turn off genes. So again, the LuxR signal sensor is really important. Now, it's not just Vibrio fischeri that has these things. So you can see these parallel systems that occur in Pseudomonas, for example. And so Pseudomonas is a really prolific producer of these autoinducers, and obviously it's a really amazing pathogen, and it's an amazing survivalist in the environment. This microbe can cause lung infections. It can hang out in the environment in the soil and cause infections and burn patients. It can cause folliculitis and jacuzzis, for God's sakes. The microbe is very prolific. But a lot of its ability to do this is predicated on quorum sensing. And so it has these parallel systems, LAS-R system, which makes a 3-oxo-C12 homoserine lactone, these little, I don't know what the hell that is, pentagons. And then the RHLI and RHLR system that you see here that makes a C4 homoserine lactone. And so the LAS system here, where LAS-R and the autoinducer combine to form a transcription regulator, activates the LAS regulon, and I'll show you a little bit about that. But this combination can also activate the RHLI and R system, and in turn that activates the RHL regulon and a variety of proteins thereof. They're downstream of that. And so here are a few of those. Again, so here's the S-adenosine methionine and acyl ACP. These are starting materials that help the autoinducer synthase, in this particular case LAS-I, and the autoinducer synthase RHLI. They take these substances and they make autoinducers with them, either the 3-oxo-C12 or the C4. And so the 3-oxo-C12 has to look for its signal receptor, the LAS-R protein, and that forms this complex. And then these guys can activate these toxins, these virulence factors. Elastase, elastase is a protease that the Pseudomonas uses to damage epithelium. For example, elastin in the lungs that helps give your lungs elasticity. The microbe can damage that. It can also degrade host defenses. So if if the host, the human body sends complement proteins or whatever after it, the bacterium can degrade those proteins. But what's also cool is that those types of proteases, like elastase, can help or increase the sensitivity of rival cells to antibiotics and stimulate their lysis. It shouldn't surprise you that they'd be competitive like that because Pseudomonas is typical in a wound. And so who else is typical in a wound but an organism like Staph aureus and Strep pyogenes and so forth. And so Pseudomonas does not want to share that wound, that delicious tissue with Staph and Strep. So it will make proteases like elastase that can increase the sensitivity of these rival cells to antibiotics and basically just weaken their cell envelope so that they're more prone to lysis. And obviously this shouldn't say endotoxin A. This article made a mistake. It should be exotoxin A. Exotoxin A. It's an enzyme that interferes with translation in eukaryotic cells, so it can kill eukaryotic cells. And obviously biofilm formation is really important. Not surprising, a microbe would need to be able to stick to tissue or to stick to the walls of a jacuzzi and protect themselves from chlorine and so forth. And the PQS is a positive regulator of biofilm as well. So these two kind of go hand in hand. So the LASAR HSL system will activate PQS, and then PQS in turn will further activate biofilm formation. And the RHL system that I mentioned as well can activate things like phenazine, and phenazines these things generate phenazine and pyocyanin they generate reactive oxygen species. So they can be used to compete against rival cells. They can make like peroxides and things like that are toxic to rivals. So pyocyanin and phenazine, and they can the RHL system. That's where the RHL comes from actually, because I think it was first discovered as a regulator of rhamnolipids, and these are like cell surface surfactants, that the microbe produces. And they can actually cause ciliostasis, which basically means the ciliated epithelium of your respiratory tract will become paralyzed. And not surprising when that happens. It's basically like what happens in cystic fibrosis patients, where the cilia don't work anymore and they can't move mucus around and the mucus builds up. And as a result, the individuals basically just suffocate in their own mucus. But that's what the rhamnolipids do. They facilitate that kind of ciliostasis and can damage the host. But they also directly kill immune system cells. So when the human body sends immune system cells like neutrophils after them, the rhamnolipids can kill them. So it's a pretty brutal combination. And so again, the microbes produce a variety of things. Like cyanide, for example, they produce cyanide as a result of the oxidation of glycine, and by making cyanides obviously a cytotoxin, so they can kill rival cells, they can kill host cells. Another cool thing that the RHL system seems to activate is a chitinase. And I guess it shouldn't surprise you if you've got a microbe like Pseudomonas that's pretty prolific in the environment, they would produce a chitinase that can help them eat, I don't know, grasshopper exoskeletons, I guess, or it probably has more to do with the potential antifungal properties, because chitin is a component of yeast cell walls. And so this is just saying about roughly the same thing, rhamnolipids and PMNs and making cyanide to damage tissue and so forth. Superoxide dismutase is it cleans up free radicals and that the microbe might encounter in the human body, and proteases that I already talked about, like elastase and alkaline protease, to destroy immune system factors and so forth. This is just an interesting little side story, but there are companies that have looked at quorum sensing as a target, as an antimicrobial target. And so these slides right here show you what I already talked about, where you have a LASI synthase making an autoinducer that's recognized by a receptor, in this case LASR, and that in turn activates gene expression. So a couple of the mechanisms I've listed here. You can create antagonists, autoinducer antagonists that look like the autoinducer, that confuse and swamp out the LASR and block gene expression. Or you can treat a patient potentially with antibodies to the quorum sensing molecules, or you can just outright degrade the autoinducers with lactanases. In this particular case, you can interfere with S-adenosylmethionine so that the synthase doesn't have a major substrate for making autoinducer, or you can interfere with some of the transcription factors that are input into the system. I didn't talk about these guys, but they're in the figure over here, but I didn't talk about them, so don't worry about that. But just, again, inhibiting factors that would result in altered quorum sensing. And then another one is antisense oligos that would interfere with, for example, LASR expression. So if you have no signal, then the autoinducer synthase is making autoinducer that's not going to do anything. This slide is an emphasis on Vibrio fischeri, the parallel quorum sensing systems in fischeri, and they're somewhat similar. Well, at least this is somewhat similar to cholera and some of the other organisms, but this one is more unique to fischeri. And so in this case, you have LuxR, the receptor, and LuxI, the product of LuxI, that's the autoinducer synthase. The autoinducer synthase, in this case, the LuxI autoinducer synthase, makes the 3-oxo-C6. Fischeri has another autoinducer synthase called AINS, and that makes a C8 molecule. And then it has a third one, the LuxS one that I already showed you with the boron, that is similar to cholera. And so again, the autoinducer 2 here feeds into this system here, the LuxPQ system. The AINS autoinducer C8 feeds in over here and competes with this guy for the LuxR receptor. It basically just, they work together. I shouldn't say they're competing. They work together where C8 first binds and primes it and gets going. That's why this arrow is thinner than this one. C8 binds first, is made quicker, and activates the LuxR system a little faster. But once you get going and produce even more of the Lux components, the 3-oxo-C6 starts to win over and becomes a stronger regulator. But in this particular case, though, it's just like I showed you before, where if you don't have any autoinducer bind, then LuxP and Q autophosphorylate, and they transfer the phosphate group to LuxU, which in turn transfers the phosphate to LuxO, which in turn activates small RNAs in HFQ to shut down LIGHT-R. When autoinducer does bind, the phosphate flow goes in the reverse direction. So there's no repression of LIGHT-R, and LIGHT-R will then be active, and LIGHT-R will in turn turn on LuxR and then feed the system. So more LuxR and so on results in more expression of the luminescence genes. And this is just a different look at that. Again, you can go through that, but here's LuxR, and then when it binds to autoinducer, and here's S-adenosylmethionine and acyl-ACP and so forth. And this is just kind of giving you the graphical appearance of how it looks. Hopefully your graphs look something like this. The graph is LU luminescence per OD as a function of OD on the x-axis. And so again, you're just looking at how intensely these guys can autoinduce in the presence of autoinducer. So the cells are obviously really can be really induced to produce excess bioluminescence. So the again, you can see how much autoinducer these guys produce. So any of the microbes in the presence of or with autoinducer producing just tons of light. The slide here is more about biochemistry of Lux gene production and what these things do. But what's really cool about this slide is that it shows you how luminescence and central metabolism are playing a tug of war. And I really wanted you to appreciate this concept, the idea that luminescence takes a lot of energy. And the question is, why would something like that evolve? Why would a microbe care about, producing light? And so we have to, and you have to put it in the context of microbe-host interactions. The microbe is making light, and the animal, in this particular case, the squid or the fish or whoever's using it, because there are lots of examples of bioluminescent symbiosis, has to find the light useful. And if the organism finds the light useful, if the higher organism finds the light useful, it will feed and house that microbe. So it behooves the microbe. There's a selective pressure to actually come up with a system like this. So it works a bit like this. So here's the central metabolism part I was talking about. So you have a reduced substrate, say glucose, for example, it's oxidized. And glucose gives up its electrons to electron carriers. And ultimately, the electron carriers, like NADH, will dump their electrons into electron transport. That generates proton motive force, and then proton motive force, of course, makes energy through, obviously diffusion through ATPases. And then oxygen is our terminal electron acceptor. Well, luminescence requires these electrons, or at least a bunch of them. And so the microbe will take these electron carriers using this Lux protein, Lux G. And so again, you can reference back to the Lux operon here, Lux C, D, A, B, E, and G. Now, the Lux G will steal electrons from NADH and transfer them to FMN, flavin mononucleotide, to make reduced flavin mononucleotide. Lux A and B is the so-called luciferase, the main enzyme here. And so the reduced flavin mononucleotide, the enzyme luciferase, and oxygen combine. And so here's the reduced flavin, here's the enzyme, and here's oxygen. The last thing that this explosive cocktail needs is the aldehyde substrate. So once these four come together, you see here, these guys rapidly come together and quickly explode into a burst of light. And they produce, in a fatty acid, they produce water, and they produce oxidized FMN as an additional byproduct. So now the microbe has to collect these things and rebuild them quickly. It's almost like packing a musket gun. you put the powder, you put the powder in, and the packing, whatever it's called, the wad that it's called, and you pack all that in there. You throw the musket ball in there, and then you fire, you shoot it. Well, that's what the microbes have to do. They have to repack their little musket gun here. And so in this particular case, the FMN has to be reduced again. The enzyme has to be recycled. The fatty acid here has to be converted back to an aldehyde using Lux E, C, and D. And so these guys are the aldehyde synthase. And so, and in that process, that takes energy. So that definitely, there's a direct output of energy here. So there's you're indirectly costing energy because you're taking electrons away from the electron transport chain, but you're directly using energy by pulling, by making aldehyde substrate. But again, you're spending energy to make a whole lot more. So here are the one-word titles for all the enzymes. And so this slide is really neat because it gives you a sense for how microbes can cross species, quorum sense, and not how, but that they do it. This was some early data that showed that Vibrio harvii and E. coli could actually talk to each other, so to speak, using the Lux S type system. And so in this particular case, what they did was they took a Vibrio harvii autoinducer mutant, and I don't know who did this, but it's so brilliant. They paint these little arrows. I don't know why they didn't need to be so sophisticated. So they paint the Vibrio harvii autoinducer mutant here, and then they have a E. coli that produces a harvii autoinducer-like molecule that they paint here and here, and then they have a negative control here. somebody who doesn't make a Harvey autoinducer-like molecule, and then they let them grow. They co-culture them. And so at this interface here, this E. coli that's producing the Harvey-like autoinducers, the autoinducer molecules are diffusing across this barrier and touching these cells here and autoinducing them, stimulating them to make a grunt load of light to the point where they're bioluminescent right at the periphery. Such a cool experiment. This slide is an additional example of that, and so in this particular, I won't get too deeply into it, but what I want you to see here is that the individuals have made in this particular research group have made a series of strains, and they're basically comparing the two sensor systems in Harvey, not comparing rather, they're testing who has autoinducers that can stimulate either sensory system in Vibrio harvii. So Vibrio harvii BB120 is the wild type, and so it has all the sensor systems, both sensor systems 1 and 2, and in this particular case, so you see BB120 has sensor system 1 and sensor system 2. BB886 has sensor system 1 but lacks sensor system 2. And BB170 has sensor system 1 but lacks sensor system 1 and has sensor system 2. And so they're basically just growing these cells, these different strains up in a flask, spinning them down, and then taking a little bit of culture supernatant and adding it to cells of either this, or this to look at induction of luminescence. And so, not surprisingly, if you grow up the wild type that has both systems and produces both kinds of autoinducer, and you take a bit of its cell-free supernatant and add it to each of these cultures, you get 100% activation of luminescence because BB120 makes both sensor system autoinducers. But if we look at, BB152, for example, we don't know what it makes. And so that's the question. What does BB152 make? And I'm not going to talk about it. I'm not going to go through this. I want you to think about what does it make and what do these data all mean? Here's Vibrio fischeri ES114, for example. And the question is, does it make sensor 1 or sensor 2 of Vibrio harvii autoinducers and so forth? So go through that and be prepared to answer some questions on the exam about it. So another element of gene expression is coupled transcription and translation. It's a way that microbes, prokaryotes in general, can be really fast at gene expression. And so here's the double-stranded DNA, and imagine RNA polymerase is moving along and transcribing. And as this transcript is coming off, the ribosomes are able to rapidly sit right down on the messenger RNA before it's even completed and start translation. So this coupled transcription and translation makes microbes fast. And that's the take-home message there. And so again, you can see the other component of this is the messenger RNA transcripts are polycistronic, meaning that there's many start and stops, many different genes encoded in single transcripts. It's not like in people where there's primarily there's a one main transcript for each protein. With bacteria, you can get a whole operon on a single transcript, but they would have different ribosome binding sites, as you can see here. So this one, there's again, a ribosome binding site, a start codon, and a stop codon. So here's an open reading frame that would encode one protein. And then another ribosome binding site, another start, and another stop. Here's a second protein. So this polycistronic mRNA encodes two separate proteins. So again, having coupling transcription and translation and having polycistronic mRNA allows microbes to be fast. The regulation of transcription elongation is really important, as I've alluded to this, and there's this mechanism. The transcription can also be regulated by controlling transcription termination, and so this mechanism that I alluded to before, that um, basically touches on the translation side, because up until now, what I've mostly talked about was regulation of the level of transcription, quorum sensing, and two-component systems, and so forth. But attenuation touches on translation as well. And so this type of regulation, called attenuation, was first demonstrated with the tryptophan operon. And so that's what I'll emphasize here. So trip operon attenuation, in addition to transcription initiation control, transcription continuation is also controlled in this operon. And so attenuation is termination of transcription within the leader region, or the leader peptide, and occurs through stem loop structures. So here we have the model for this. And actually I can oops. So here's the TRIP-L leader. And so we've got regions 1 and 2 and regions 3 and 4. So here's the RNA that's being made. So imagine that this is the operon for the biosynthesis of tryptophan. Now, obviously, the cells don't need biosynthesis proteins for tryptophan if there's a ton of tryptophan in the medium. So not surprisingly, the presence of tryptophan will stop this from happening, whereas the absence of tryptophan will encourage this expression of the tryptophan operon. So there's something called the TRIP leader, and then TRIP E, which is, again, a major protein for biosynthesis of tryptophan. So, oops. So as I say, there's four regions involved: region 1, 2, 3, and 4. They're numbered relative to the 5' end of the transcript, with 1 closest to the 5' end. Region 2 is complementary to both regions 1 and 3. So this is region 2 here. In this form, you see it binding to region 1, but it's also complementary to region 3 here. So region 2 can actually form a loop with either 2 and 3 or 1 and 2. Similarly, region 3 is complementary to both regions 2 and 4. So that's pretty self-evident. When regions 1 and 2 form a stem loop, as you see here, region 3 forms a stem loop with region 4. Immediately following region 4 is a uracil-rich region called the attenuator. So that's down here. When the region 3-4 stem loop is present, region 3-4 stem loop is present, this guy here, it acts with the attenuator to form a termination for transcription at the end of TRIP-L. So basically what that means is that the RNA polymerase, as it's moving along and laying this down, this little wrinkle here will kick the polymerase off. And so it'll basically terminate transcription. When region 2-3 stem loop is present, the terminator structure does not form, and transcription proceeds through TRIP-E, the main biosynthesis protein, and other tryptophan biosynthesis genes in the operon. So there are 3 scenarios possible with respect to attenuation in the TRIP operon. So here's our mRNA for the tryptophan leader. Here's polymerase coming through. So in that form, region 1 and 2, region 3 and 4 stem loop forms, and transcription is terminated at the end of the TRIP-L gene. The other 2 scenarios involve translation coupled with transcription. The difference between these 2 scenarios depends on whether or not the ribosome gets stalled in region 1. So let's take a look. Oops. If tryptophan levels in the cell are low, as I talked about, then tryptophan transfer RNA levels will also be low. So in that 2 adjacent tryptophan codons in region 1 determine whether or not stalling will take place. So here's our guy. This will cause the ribosome to pause at the 2 tryptophan codons as it waits for a tryptophan transfer RNA. See that? So again, the A site that you see here is waiting for a tryptophanol tRNA. And if it has to wait too long, that allows the termination stem loop to form. This will cause the ribosome to pause at the 2 tryptophan codons as it waits for the transfer RNA. Allowing 2 and 3 to form a stem loop. This prevents the formation of the 3-4 stem loop. Transcription is allowed to proceed. So that should make sense, right? That the ribosome stalls, suggesting that the cell is starved for tryptophan because the ribosome isn't moving quick enough. That allows this anti-terminator stem loop to form and allows the polymerase to continue reading through. Oops. If tryptophan levels in the cell are high, then everything moves quickly. The 3-4 stem loop readily forms, leading to termination of transcription. So that just kicks the 3-4 actually kicks the polymerase off and blocks the transcription. And so you can kind of follow this diagram. It's basically just paraphrasing everything. I just paraphrased everything that in that image, that movie rather, right on up to, 3-4 forming. And because, again, there's no pause here. Tryptophan levels are high, no pause, 3-4 forms. And that's the terminator loop forms and kicks this guy off. So you don't get trippy. Conversely, the 2-3 forms here, the so-called anti-terminator loop, because this guy's stalled. So anyway, that's the overview of the attenuation process.